student, this is the last topic of unit 3, and in this topic, in this lecture we have to cover the topic synthesis of 5-hydroxytryptamine, melatonin, dopamine, noradrenaline and adrenaline synthesis with the catabolism of the heme. So, in this lecture we have to discuss about how to synthesize these neurotransmitters and what are the significance or biological importance of these neurotransmitters and how to how catabolism of heme occurs. So, we have to start with the synthesis of serotonin. What is serotonin and how they affect? Serotonin is an important chemical and neurotransmitter. It is present in our human body. It is synthesized in the brain. So, it is responsible for normally for the neuron functioning, and it is helpful to regulate mood and social behavior. Serotonin is generally responsible how to regulate your social behavior, how to talk to person, and it is also responsible for maintaining appetite and digestion. It is also helpful for the sleep and memory. So, serotonin shows in such conditions. So, those basically work done by the brain is basically affect with the serotonin. Serotonin synthesized in brain and transported to the body, and how it is synthesized. So, this is the pathway. There is tryptophan. So, serotonin or 5-hydroxytryptophan is synthesized by the tryptophan. Tryptophan is having the indole in their structure. So, there is indole ring in this structure present, and in this indole there is propionic acid present in this structure with the presence of one amino group. So, tryptophan is an amino acid, and this tryptophan is reacted with the or catalyzed by the tryptophan hydrolyase enzyme or tryptophan 5 monooxidase enzyme to form 5-hydroxytryptophan. This 5-hydroxytryptophan is having the hydroxyl group in this position. So, there is 2 hydroxyl group present here. So, this is known as 5-hydroxytryptophan, and this 5-hydroxytryptophan is when reacted with the 5-hydroxytryptophan decarboxylase enzyme, there is removal of this carboxylic group from this reaction, and there is a removal of one carbon dioxide group from this, and there is formation of serotonin. So, what is the difference between the structure of tryptophan and serotonin? There is removal of carbon जो है C double bond OH group या carboxylic group from this tryptophan to form the serotonin here. Okay. So, what is the significance of this serotonin? And this serotonin is basically improves appetite and digestion. Serotonin helps in increasing appetite, and at the condition where the patient having the too much efficiency to eat. So, at that condition they can control appetite and improve digestion also. This serotonin work on the as an vasoconstrictor, and it is helpful in the smooth construction of the muscles. So, serotonin help in the normally construction of the smooth muscles, so that the it help in the walk or locomotor activity also. This serotonin helps in the mood and memory, because it is synthesized in brain and it is helpful for the normal activity of brain. So, this serotonin improves our mood and our memory. So, if there is any deficiency of serotonin occurs in our body due to the less presence of tryptophan or conversion of tryptophan to serotonin, they can affect our mood and memory. It can affect also our memory conditions, and serotonin is also helpful to relieve pain. Whenever there is pain present in our body, serotonin help to improve that pain, and it also helpful for the blood clotting controlling. It serotonin affect the different factors which are helpful for the blood clotting, and they stimulate that factors to increase the blood clotting, and help in the wound healing. That is why it's help in the wound healing. So that because the main nature of serotonin is the vasoconstriction. So by blood clotting or wound healing, they can constrict the smooth construction of muscles, so that the wound or clot condition or that wound particular shows constriction, so that it's controlling blood flow and it controlling wound healing or help in wound healing. It improves sleep and memory. Sleep and memory, because it is also related to the brain. So, brain whenever brain needs relaxing condition, so it improves the sleep of us, and it is also helpful in the memory. The second thing is biosynthesis of melatonin. How melatonin is synthesized in our body. So, this is the pathway of synthesis of melatonin, and this is also tryptophan is precursor for this melatonin, and this tryptophan is responsible for the synthesis of melatonin here. So, tryptophan, we already discussed the structure of tryptophan. There is indole moiety present here, and there is presence of propionic acid in this indole structure, and when this tryptophan hydrolyzes is reacted with the tryptophan 5 monooxidase enzyme. So, there is formation of 5 hydroxytryptophan, and this 5 hydroxytryptophan is when reacted with the 5-HT decarboxylase enzyme to removal of the carboxylate group here. So, there is formation of serotonin. We have already learned about the how to synthesize or how to prepare serotonin from the tryptophan. So, after synthesis of serotonin, this serotonin 9 acetyl transferase enzyme is responsible for the conversion of this serotonin to N-acetyl serotonin. This N-acetyl serotonin, with the help of this serotonin N-acetyl transferase, there is one N-acetyl group is also attached in this serotonin enzyme, and this hydroxyindole orthomethyl transferase enzyme is required in this reaction, and there is formation of melatonin. So, there is formation of melatonin here. So, there is attachment of methyl group in this reaction or this structure. So, the complete C double bond OH group is present here, and indole group is already present, but there is attachment of methyl group in this structure. Okay. So, this is melatonin, and what is melatonin and what is the biological importance of melatonin, we have discussed now. So, what is melatonin? Melatonin is showing the important role in the regulation of sleep cycle. We all know circadian rhythm, that particular rhythm is responsible for the our sleeping and opening of eyes. So, this circadian rhythm is maintaining the regulation of our sleep cycle. It occurs in every 24 hours. So, melatonin plays an important role in the regulation of our sleep cycle, and it whenever this is whenever there is production of melatonin reduces in our body, this is affect our sleep pattern or sleep cycle. So, melatonin is generally released from the pineal glands of brain during the night times or night hours. So, whenever in the night times it occurs, it synthesized in the brain, that can show its sleep cycle or circadian rhythm. So, our mind feel that we have to sleep. So, this melatonin is controlling our sleep cycle or awakening cycle. So, this occurs in every 24 hours in the night time, so that it can release from the brain by the pineal gland, and it control or regulate our sleep cycle. It also induce the physiological changes, it also induce a physiological improvement to promote sleep, to control sleep. So, that it can also control or decrease body temperature and respiration rate. At the time of physiological changes or at the time of sleep condition or sleep cycle, circadian cycle, at that time our body temperature and respiration rate is also decreased by these melatonin, so that a person can easily take sleep and there is no other pressure occurs in our body, so that it can cause a physiological change to control our temperature and respiration rate. And it is also helpful in controlling insomnia. As usual, we all know that it's controlling the sleep cycle, and it controls the circadian rhythm of our body. So it helpful in the controlling the insomnia in children and adolescent, those kids having the age of 2 to 18. So in such situation, it control the autism spectrum disorder. What is autism spectrum disorder? The patient which are having the inability to talk with patient, or controlling their brain to use their brain to talk to someone, or thinking about anything. So this melatonin help in patient. Autism is that condition in which a kid or a adolescent is not properly working, or they are not properly think, they are not properly talk with anyone, and they are showing some reducing in their growth condition. Growth occurs in such kids or adolescent. So this melatonin control the autism spectrum disorder. Okay. Next is dopamine. How to this dopamine is synthesized, biosynthesized in our biological system. It is helpful. The precursor of the dopamine synthesis is phenylalanine. This phenylalanine is the main precursor, or this phenylalanine is having the aromatic ring in their structure, having nitrogen amino group and carboxyl group in their structure. And if we have we have seen that phenylalanine hydroxylase enzyme reacted in this reaction with this phenylalanine to form tyrosine, tyrosine here, and this tyrosine having the hydroxyl group in the para position. So the difference between the phenylalanine and tyrosine, there is presence of hydroxyl group at the para position of aromatic ring. Okay. So phenylalanine is converted into the tyrosine by phenylalanine hydroxylase enzyme, and this tyrosine is again converted into the L-dopa by the help of tyrosine hydroxylase enzyme. So there is presence of or addition of one hydroxyl group at meta position, because meta position. So with the help of this tyrosine hydroxylase enzyme, so there are presence of two different hydroxyl group in this aromatic ring. The other side chain is similar, that is not having any change in that side chain which is present in the phenylalanine, tyrosine and dopa here. So this there is conversion of this dopa to dopamine by the changing of this carboxylase enzyme here, sorry, carboxylase group here, because in the whole structure there is no change in the side chain of this phenylalanine. Thus there is change of addition of one hydroxyl group here, addition of one hydroxyl group in the third step. So there is two hydroxyl group present in the dopa here. So in that aromatic amino acid decarboxylase enzyme, there is removal of carbon dioxide here, or removal of carboxylase group here. So there is formation of CH2, CH2, NH2, that is dopamine. So there is two hydroxyl group present here, and there is CH2, CH2, NH2 group present in formation of this dopamine. Okay. So what is the significance of dopamine? Dopamine, what is dopamine? Dopamine belongs to the catecholamine. Catecholamine are neurotransmitters, and dopamine is a precursor for the synthesis of adrenaline or noradrenaline, because when dopamine is synthesized from the phenylalanine, this dopamine work for the formation of adrenaline and noradrenaline, and these are synthesized from the dopamine. So these dopamine plays a significant role in the learning, goal directed behavior, controlling your brain, use of your brain, motor control, hormone control, formation or synthesis of various hormones, control flow of information from other areas of the brain, because basically dopamine is a precursor for the formation of adrenaline, noradrenaline. So this can control the other areas of the brain, and they can affect the learning and goal directed disorders, and they can also affect the goal disorders, motor control and regulation of hormones. So it dopamine can control also movement and nerve control, because dopamine present in the brain, and they can affect or they can control the nerve movement, nerve formation, movement condition. So that dopamine is very important. It shows a very important characteristics for the formation of other neurotransmitters like adrenaline and noradrenaline. And what is this biosynthesis of adrenaline? We all knows that adrenaline is synthesized from the dopamine. So just we have to discuss about this pathway. Okay, so tyrosine, we have already knows that tyrosine is converted into the levodopa to dopamine, which we have already discussed in the previous slide. So we have to start with this dopamine here. In dopamine, there is 2 hydroxyl group present in the aromatic ring, and there is CH2, CH2, NH2. Okay, this is a side chain. So in dopamine is when reacted with this beta hydroxylase enzyme here, and there is formation of norepinephrine, which is also known as noradrenaline. This norepinephrine or noradrenaline is when reacted with the N-methyl transferase in adrenal medulla. So this reaction occurs in the adrenal medulla with the catabolism of N-acetyl transferase enzyme, and there is formation of epinephrine, which is also known as adrenaline. This is a synonym of adrenaline is epinephrine, and synonym of noradrenaline is norepinephrine. Okay. So what is the difference in the structure? There is presence of NHCH3 group here. So there is presence of one methyl group in this structure, and there is presence of one hydroxyl group in this structure also. So difference between the norepinephrine and epinephrine is presence of methyl group in adrenaline, and absence of methyl group is known as norepinephrine. Presence of methyl group with the amino or nitrogen here is known as adrenaline or epinephrine. Okay. Thick. And what is the significance of this adrenaline here? So we all knows that adrenaline is produced in the adrenal gland, while noradrenaline is secreted in the certain neurons of the brain as well as adrenal glands. Difference between the noradrenaline and adrenaline is the in their structure, because adrenaline having methyl group in their structure and noradrenaline does not have methyl group, okay, that having the amino group in their structure, and adrenaline produced in the adrenal gland, but noradrenaline is also secreted from the certain neurons of the brain as well as adrenaline adrenal gland. Both are the example of catecholamine, and these catecholamines are main neurotransmitters of sympathetic nerves. Sympathetic nerves in the cardiovascular system. So these noradrenaline and adrenalines work as neurotransmitters of sympathetic nerves, and because of presence of or synthesis of adrenaline, which is the main hormone of adrenal medulla, this may cause to increase or maintain blood pressure. The main work of noradrenaline is used to increase or maintain blood pressure in acute situations like minor heart attack or heart attacks or situation, cardiac arrest situation. So in that situation, there is there any problem in cardiovascular system? Noradrenaline is generally used for the increasing or maintaining the blood pressure, and adrenaline used to generally treat the low blood pressure. Adrenaline is work to increasing the blood pressure. It can give the pressure or tension to the blood so that it can flow in the at the fast rate, and increasing the blood pressure associated with the septic shock. At that condition, is there any shock occurs in our body, or any allergic reactions. In such situation, when our blood pressure is going down, in that situation adrenaline is released from the body, or it is synthesized from the body, and it shows to control the reducing blood pressure or hypotension. Okay. Next is catabolism of heme, how hemoglobin or heme is catabolized in the body. So, heme, what is heme? Heme is a iron or iron coordinated compound which is having porphyrin system or porphyrin ring of hemoglobin, and it is act as a ligand. Heme, what is heme? Heme is a non-protein part of the hemoglobin, or globin is a protein part of the hemoglobin. So, most important compound, hemoglobin is the most important porphyrin containing compound, and porphyrin ring जो है cyclic compounds है which is having a pyrrole ring in their structure, or which is controlling or held together by the methynyl bridge, which is having methynyl bridge in their structure. 4 pyrrole ring is present which are connected together or held together by the methynyl bridge. It is synthesized in the liver and bone marrow. So, heme is always synthesized in the liver and the bone marrow. So, how to fade down, how to broken down of this heme. So, this heme is broken down in macrophages of the reticuloendothelial system of the spleen and the liver. So, this occurs, catabolism of heme occurs in the spleen and liver, and whenever there is when RBCs are going to be the fade down after 120 days, because 120 days is the normal period of RBCs working. So, when after the RBCs are after 120 days RBCs are matured enough to degraded by the reticuloendothelial cell or reticuloendothelial system, mainly in liver and spleen. Firstly, hemoglobin is dissociated into the two part, heme and globin, and this heme can be this globin can be reutilized in for the formation of hemoglobin again, or it is can be degraded by the individual amino acid. Those amino acid are present in that particular globin, that it is degraded into the particular amino acid, and the amino acid then undergoes into the metabolism is also, and it is also participate in the fresh globin synthesis. So, heme and globin both are those are departed into two different parts, and globin is also converted into the helpful in the conversion of different new hemoglobin or degraded into the different amino acid. By amino acid they can synthesize again. So, the catabolism of heme is also carried out in the small fraction of cells by heme oxygenase enzyme, which is occurs in the presence of this is controlled in by the presence of NADPH enzyme, NADPH and oxygen, which ferric acid, carbon monoxide and green pigment, which is also known as biliverdin. This biliverdin is released as biliverdin, and when this biliverdin is converted into the bilirubin by red orange pigment, this biliverdin is converted into the bilirubin, and this bilirubin and biliverdin both are known as bile pigments. These bile pigments or bilirubin or biliverdin are the excretory product of the heme. These daily bilirubin formation in the body is around 250 to 350 mg per deciliter, and this is yielded not only by the hemoglobin, but from the ineffective erythropoiesis, mean the formation or synthesis of RBCs in the body, ineffective synthesis of RBCs, and also from the other heme protein like cytochrome P450. Okay. This is the general pathway. Hemoglobin is synthesized again transfer into the heme and globin here. This globin is entered into the amino acid pool to build new protein, and heme is again converted into iron and ferritin here to form the transported form of this iron, and porphyrin is converted by the hemoxynase enzyme into the biliverdin, which is again biliverdin reductase is reacted in this reaction to unconjugated or indirect bilirubin here to form the different bile acids here, bilirubin, urobilirubin, and this particular bilirubin or biliverdin is excreted by the from body by a stool or urine. Okay. And if there is any disorder occurs due to the abnormality in the removal of heme from the body, they can cause jaundice. Jaundice is a condition where the bilirubin concentration in the blood is increases, which cause the yellow discoloration of eyes and skin, which represent the increased concentration of serum bilirubin. And newborn having the low level of bilirubin, those kids or newborn which are having they are normally having the high rate of jaundice in because the mother having the formation of different RBCs, and when they are taking birth, after that their liver is not mature enough for the formation or catabolism of these heme here. So, they are showing the presence of bilirubin and biliverdin in the blood increases. That is why every kid, every newborn having the small amount of jaundice in at the time of their birth. So, jaundice is a clinical condition characterized by the presence of yellow color of yellow color at the sclera of the eyes and the skin. This is due to the immature liver present of that newborn, and by the days passes this can be controlled itself, or it can be controlled by the medicines also. So, this is of two type: hemolytic jaundice, which is jaundice basically associated with the increased hemolysis of erythrocytes, and the second one is hepatic or hepatocellular jaundice. This is caused by the dysfunction of the liver due to the damage of the parenchymal cells. So, this is due to the increase or level of conjugated or unconjugated bilirubin in the serum, and having the dark color urine, increased activity of alanine transaminase enzyme and aspartate transaminase enzyme, which is circulated which is having high amount in circulation due to damage of the erythrocytes. So, this is also a obstructive regurgitation jaundice. This is due to the presence of bile acid prevents the passage of the intestine. Okay. This is all over the today's class and unit 3rd. So, this is completed unit 3rd here. So, thank you.